UV light. Okay. Give our hand. Oh, wait, how do I use this?
Just click like
If light. 71% of the Earth is covered in water. 97% of that water is undrinkable. 2% of drinkable water is locked up in glaciers. 1% of water on Earth is used to fulfill all of humanity's needs. So, my paper is titled Rapid Water Disinfection Using Vertically Aligned MOS2 Nanofilms and Visible Light. Or more simply put, rapid disinfection of water using light. So, why this paper is important? We need water. Every living thing, including humans, plants, and animals, needs water to survive. And more importantly, we need clean water. Clean water is really expensive to get and even harder to distribute. Scarcity of clean water in the world is a really prominent global challenge. So, my paper is very chemistry intensive, so I'm going to give a little bit of background. You'll hear me refer to Bandgap a lot, and what that basically is many of you will remember from Chem 1 that in order for an electron to move up an energy level, it has to receive some form of energy, and in this case, a photon. So the bandgap is the space in between the valence band and the conduction band, where no electron exists. The important thing is that it needs to receive the appropriate amount of energy to go from the valence band to the conduction band, which is only found in a certain range of photons of light. Are there any questions on that? Okay. Next, I'll refer to photocatalyst a lot, and what that is basically what it sounds like. It's a catalyst, a substance that speeds up the rate of reaction for photons. So it allows photons to be absorbed more rapidly. And since we're getting our energy from photons, this is essential to harvesting their energy. And most photocatalysts are semiconductors, especially in the case of my paper. I'll talk about MOS2, molybdenum disulfide, but TiO2 is also a common photocatalyst, but I'll explain the differences between those a little bit later. Okay. Last bit of background, I'm sure many of you have seen the light spectrum, but it's important because it has tons of different kinds of light and wavelengths. But what I'll be focusing on is UV, visible, and infrared light. So visible light, most importantly, is between the 400 and 700 wavelength. So just keep that in mind. Okay, so my paper focuses on using sunlight, which is really smart because sunlight is available everywhere, every day, and it's cheap. I don't think any of us have to pay to look at the sun. And it's composed of visible light, about 50%, ultraviolet light, about 4%, and infrared light, about 46%. And there are two main ways that sunlight interacts with the photocatalyst to disinfect water. First is directly through UVA, but that can take anywhere from 6 to 48 hours, which is a really long time to wait for water. The next is indirectly through ROS, or reactive oxygen species. So ROS is an altered form of oxygen that is highly reactive and is involved in a lot of reactions. It's created by metabolizing oxygen, and as I hope most of you know, oxygen is in water, H2O. So ROS, the more light that is exposed to the water and the photocatalyst, the more ROS will be generated. And what ROS does is it goes and reacts with the molecules in the water to deactivate them. Are there any questions on ROS? Okay. So here's a general process overview of my whole paper. So the first is where we get the energy from, the sun. And then the thing that harvests the energy is the MOS2, the photocatalyst. Then the photocatalyst takes that energy and creates electrons, or electron hole pairs. And as you can see, they're armed and ready to fight the pathogens and bacteria. Then the electrons go and they interact with the water and oxygen, because it's made of the reactive oxygen species. So then once they react, they create ROS. And then the ROS goes and damages macromolecules and pathogens in the water, and then we get clean water. Are there any questions on the process? Okay. So, how it works. MOS2 is this chip, and it's about half the size of a postage stamp. It's extremely cheap and easy to produce, especially in relation to other forms of water purification. Okay, so then this picture on the left basically shows the whole process overview, with the sunlight hitting the chip, creating electron hole pairs, and then reacting with oxygen and water, and then killing the bacteria. So, and then also these block-like looking things, layers on the MLS2 chip, this is an actual picture of them. As you can see, there's like a lot of little layers, especially for something like that. Okay, so can anyone tell me what the first step in the process overview is? Where does the energy come from? Yes.
The sun.
Yes, correct. Can anyone tell me what absorbs the energy? Yes.
Photocatalyst.
Yes, the photocatalyst, MLS2. And then what does harnessing that energy then create?
Electrons.
Electrons, nice. And then the electrons go and interact with what?
The H2O and the oxygen.
Nice. And then both of those create Allie?
The enzyme.
The fighty thing.
Close, yeah, the ROS. Yeah, so that's the one doing the actual disinfecting. And then the ROS goes and does what? Kills all the bacteria. And then we get clean water. Okay, nice. That was impressive. Okay. So, the first thing the scientists wanted to test was the absorption of the MLS2 and the sunlight based on its wavelength. So, as you can see from the graph, it absorbed all the wavelengths from 400 to about 800. And that's important because visible light, as you can see on the picture on the left, is 400 to about 700. So, from this we can say that because sunlight is made of 50% visible light, this MLS2 is able to use a lot of that light, as opposed to like ultraviolet light or infrared light, which would be over here. But visible light is the majority of sunlight, so this is really good. Okay. The next thing they wanted to test was what was actually doing the disinfecting. Was it the light, was it the MLS2, or was it a combination? So the black line on top is just light on water, with nothing else. And if you've ever tried just having light on water, it doesn't really disinfect anything. It just sort of stays the same. The next thing they did was they put MLS2 in water in a completely dark setting. And that actually had a little bit of result, but still not enough, not so much that the water is clean enough to drink. But the green line is combining light with MLS2. And that achieved over 99.999% disinfection in about 2 hours. So that's a really big step. Now we know what's doing the work and what's required to achieve full disinfection. So, the next thing they wanted to test was the thickness of the MLS2 and what the ideal sort of characteristics of it were. So when I say the thickness, I mean the thickness of like these blocks and the layers aligned on top of the chip. Okay, and then the first line, the black line on top, is really, really thick layers of MLS2. They wanted to see if that would have a better effect on disinfection. But as you can see, it kind of stayed relatively flat and didn't really disinfect the water. The blue line is actually, I'll come back to the blue line. The green line is really thin layers of MLS2, which showed disinfection in 120 minutes. So that was really good. The blue line, they actually wanted to take the vertically aligned layers and align them horizontally. But, so that kind of worked, but it still didn't really get total disinfection. The reasoning behind this is if you think about the vertically aligned layers as like a dish of water that you're trying to evaporate. Because it's like a big pan or dish, there's a lot of surface area exposed, so more of the water will evaporate. Where if you put the same water in like a really tall cup, that water would take a really long time to evaporate because only the surface can evaporate. So that's the same idea. There's more active sites if it's thinner and vertically aligned.
What's the y-axis?
The y-axis is the disinfection. So that right there, that's like 100% disinfection. Yeah. Okay. Next thing they wanted to test was how different lights affect the MLS2 and what kind of lights are ideal for disinfecting water. The red line is red pass filter, which is just infrared light. And if you remember from the sunlight slide, infrared light is about 46% of sunlight. And then the blue line is UVF, which is a combination of infrared light and visible light. So, from this graph, you can see that the infrared light wasn't absorbed until about its wavelength started, around 700, 800. But that means none of the visible light was affected, or absorbed, or used. The UVF, on the other hand, was the combination of visible light and infrared light, and because of this, a lot was absorbed in the lower wavelength ranges, and a lot of that energy was able to be used. And then from the graph on the right, you can see the disinfection results from just using the infrared, very little, and then the infrared and visible light achieved total disinfection in 2 hours. Any questions so far? Okay. So I mentioned TiO2 before as another photocatalyst, but MLS2 was used in this paper because of how they proved that it worked better than TiO2. In the lines with the circles, the top two lines, they had it under UVF light, which was the combination of visible light and infrared light. And that worked pretty well for TiO2, getting a similar result, disinfection in 120 minutes, and MLS2, or TiO2 didn't really work at all. But then the next thing they tested was MLS2 and TiO2 on sunlight, and sunlight is visible light, infrared light, and UV light. And MLS2 showed a much faster disinfection in only 60 minutes, where TiO2 didn't even reach disinfection. So now our best time and best disinfection is MLS2 in 60 minutes instead of 120 minutes. Okay. The last thing they wanted to do was test the different types of catalysts that could affect the rate of disinfection for MLS2. So the blue line is just the MLS2 on its own, and this is all under UVF light, so we're going back to the 120 minutes. But on its own, it's at 2 hours still. And then the green line is when we used gold as a catalyst on top of the MLS2, and that achieved disinfection in 60 minutes. And then the orange line is when we put copper on the MLS2, and that proved to be the best catalyst available, achieving disinfection in only 20 minutes. And if any of you have taken Chem 2, you'll know that in a lot of redox reactions, we use copper and zinc and stuff like that, but it's important that copper was used because it's really good at oxidizing and is involved in a lot of redox reactions, which is what ROS is. Okay, so we concluded then that copper with MLS2 was the most effective because it worked faster and better than TiO2, and it was the most effective catalyst to MLS2. And MLS2 is better than TiO2 because it can absorb visible light, infrared light, and UV light. And MLS2 was tested on a variety of different bacteria, and they all were disinfected, so that's a really good sign. The next steps for this technology is testing. So first we need to test the MLS2 with different catalysts and maybe different ranges and different widths of the layers. And we need to test it on a lot of different kinds of bacteria. There are thousands of types of bacteria out there. And even in other things that can affect water, like viruses and all of that. So we need to test it on different kinds of things and in different settings. All the water used in this trial was made in a lab, or was from a lab. So we need to test this on salt water and fresh water and all different kinds of water infected with different things. Next, we need to figure out a way to produce this and make it available to the world. This technology is really new, and if funded properly and made available, it can have the impact to change the world. Education. We need to make sure that people know about this technology. It's a very recent study. It was published in the last year. And honestly, I didn't know anything about this before this paper. So we need to make sure people start hearing about this and developing ways to use this, and especially people in developing countries who need this, and this could be the difference between life and death for them. They really need to be educated and know that this kind of technology exists. Lastly, we need to distribute it. It's kind of good if we have a technology that works, but it's useless if we can't get it to the people who need it. So the most important thing I think of these steps is to get it to the people who need it and get people who maybe don't need it, getting them to promote it and send it out and bring it to people who need it. Thanks. Any questions? Yes.
So how much water, so you showed the little chip, so how much water in that 20 minutes can it disinfect?
They didn't really talk about that in the paper, but the way the chip works is that it's placed in like a relatively small amount of water. It couldn't be like placed in like a big pool and disinfect the whole pool. I think it more so works on like a smaller scale.
Did they specify like where they got the water?
Yeah, so all the water was from the lab, and they made all the water in the lab. So it was controlled. Yeah. Oh yeah.
Do you think there'd be any potential concerns or health effects for later on once you've killed the bacteria that are still reactive oxygen species, or is that taken care of?
So when the oxygen goes and disinfects all the bacteria and pathogens, it doesn't necessarily like filter them out so that they stay in the water. It actually just deactivates them.
Right.
So in the sample where they used copper as a catalyst, there was no copper detected in the water after being disinfected. So it's very safe to drink.
Next week, I'm presenting, so come out and watch me.